there is this booklet of mine, Teaching on Evolution in European Schools, which is available, and anyone who wants, you can take as many copies as are available here, so you're welcome to help yourself to it. And basically what I'm going to say is contained in that booklet. Now, as Peter had just mentioned, I am a forester by education. I'm a biologist. My own speciality is population genetics, but it's populations of forest trees. Now, trees grow long time, and my experiments are of a very special type that are not producing very fast results that would be useful in combating the theory of evolution. However, I have been also a teacher, academic teacher, and I was teaching population genetics to biology students, not only to forest students, but also to biology students. And it is during that time that my own children have reached secondary school, and I have found that in the textbooks, school textbooks that my children are using, there are things which I could not accept as a population geneticist. Now, when I used to go to school, evolution was taught on the basis of paleontology. In fact, I'm from that generation where my school textbook, there was the Spildon skull was still there. So it was a different time when I was studying at school, and I assumed that the paleontologists have it all settled, and we know everything about evolution from the paleontological research. And then I realized from the textbooks of my children that the main evidence for evolution comes from population genetics. And here I am teaching population genetics to biology students, and I didn't know that population genetics is providing evidence for evolution. Now, what is shown in the school textbooks is that we can demonstrate that races are forming in nature. Indeed, we can. We can also use that knowledge in breeding work. We produce races ourselves. So this is well documented that races form naturally and they can be formed artificially. Also, the school textbooks say that race formation can be defined as microevolution. Okay, it's just a word. It doesn't change the nature of the phenomenon. But then comes the conclusion. Microevolution exemplifies a small step in macroevolution. And this is explained in the textbooks as a very important conclusion from all the research that is available on race formation. That this is a small step in evolution. We can demonstrate evolution on an example of a small step. This small step being race formation. Now, the other thing that is in the school textbooks is that mutations are a source of new genetic variation. Of course, if we have new things, and evolution presupposes that there is advance in complexity, so the new things are obtained thanks to mutations. And when it comes to showing an example of a positive mutation, what the school textbooks give is that races resistant to man-made chemicals are examples of positive mutations. man-made chemicals are things we use as pesticides, herbicides, insecticides, and so on. And then there are organisms that develop resistance to these man-made chemicals. Obviously, something that did not exist before the chemical was produced. And now the plants or the animals or the bacteria or whatever are developing a resistance to this man-made chemical. So here is an example of a positive mutation. Well, as a population geneticist, I question the postulate 3 and postulate 5. The postulate that microevolution is an example of a small step in macroevolution. And I question the claim that races resistant to man-made chemicals are examples of positive mutations. Now, I shall try to explain why I object to this way of presenting information about evolution in school textbooks. And I've checked. This is the same. It's not only in Poland that we have this in the school textbooks. It's in other countries also. It's the same arguments, the same examples. It's all over the place. Now, this indicates that the actual explanation of evolution that is given to children is based on these two postulates, which I find unacceptable. And I'll try and show in a small example, in a simple manner, why I object to these postulates. Now let's start with the population. A variable population, and this is really what we observe in nature. Populations are variable. Here we have in this room, I don't know, about 50 people. Each one of us is different. We're all the same species, but we are different. And the more we study populations, the more we find that there are individual differences. Okay, so this is something that exists. It's a fact. There is variation within populations. Now, if we, for some reason, get a fragment of the population isolated, and here are shown two examples where fragments of that population have been taken out and placed in some other condition. Now, what do we see from this isolation? The isolated fragment does not represent the whole population. There are some types which are absent in the isolated fragment. It has lower diversity. It is less variable. So, for example, here, this fragment here, which I've isolated, happens to have the white variant. Now, this fragment, isolated, has no white variant. This one happens to have the magenta one and the heavy green. This one does not have the magenta or the heavy green. So already the two isolates are different from the very fact that they are isolates as small samples. And isolation by itself creates differences. These two populations are different. Their genetic diversity is lower than the genetic diversity of the whole population. Quite by accident. They are different. Isolated populations usually vary in size. At each generation, there may be more or less individuals because they have been placed in a somewhat different environment. And the reduction in size of the population continues to cause a reduction in the variability. And of course, as long as the population is isolated, it is the generation at which it is smallest in size that will determine the extent of this reduction of variation. Furthermore, when we have a small population, we will get inbreeding. There will be crossings among relatives. Now, when we have relatives crossing, we get a process which is referred to as genetic drift. Inbreeding leads to reduction of genetic information due to what is known as homogenization. The two elements we are inheriting from parents may be the same because the parents are related. So gradually we are getting less and less variation within each individual. So inbreeding leads to accidental loss of genes. And this is a process we know. It exists in nature, it has been studied. This happens. At some stage, these small populations may suddenly start growing because the conditions become favorable. So we'll get more individuals, but only on the basis of the variation that exists in that population when it was small. And these two populations, even if they grow, they will continue to be different. So we have genetic differences between these two populations from genetic drift alone. It's just a natural, accidental reduction of genetic variation that will lead to differences between races. This is how races form. Now, Darwin was a very good observer, and he studied bird finches on the Galapagos Islands, and he found that on the different islands they have different beaks. Now, obviously, at some stage in the past, the birds were separated into separate populations on these islands, and for some reasons they have developed different features. So these Galapagos finches, they have different beaks. They are related, they are sexually compatible, but they do not interbreed because they are on separate islands. Now, the differentiation comes from genetic drift. Darwin was a good observer. He noticed this, but his explanation was different to what we have available now. The explanation that we can use now shows that this could have these differences could have arisen from genetic drift. Now let's go back to these two populations, which are different. There's another element I haven't mentioned yet, but Darwin has mentioned it, and it is at the basis of his theory, is that there is such a thing as natural selection. That in different environments, different features are suitable, and some features are unsuited for the particular environment. So we will get natural selection because of the environmental differences. Now let us imagine that from this population, we have selection against blue. So that's what we'll get without the blue. And if in this population we get a selection against red, this is what we'll get, a race without red. So the differences between the races grow. These are again natural races. This happens. This happens when the isolated fragments of the initial population find themselves in different environmental conditions, because it's the environment that does the selection. So we have natural races differing quite substantially from a natural process that is based on elimination of fragment of the variability. So as you see, in these two populations, we are getting greater and greater differences due to natural processes that reduce the genetic variation. They reduce the genetic content. They reduce diversity. They are less and less variable. Now, what do we find in the school textbooks? There is a moth called Bistona betullaria that stays on the bark of birch. And when the bark of birch is white, it's this type of moth that dominates. But in industrial regions where the bark was soot covered due to pollution, the dominant moths were black. And this is in the school textbooks explaining that here we have evolution in progress. We have a change of environment due to pollution. There is a selecting agent, which are birds. And the birds see the white or the gray moth on a black bark, so they eat it. And they see the black moth on a white bark, so they eat it. But if they don't see the moth survives. So here is a selecting agent, the bird, that will leave the black moths in regions where the bark is black, and will leave the gray moths in the region where the bark is white. This is given in school textbooks throughout Europe and the United States as an example of evolution in progress. The reason why these moths differ, this population differs from this population, comes from isolation, genetic drift, and natural selection, all natural processes, all based on reduction of genetic information. Now let's look at the situation where these isolated populations cease to be isolated. And this is something we observe when separate ranges of distribution of a species meet. And we get a richer gene pool. We speak of introgression. We speak of hybrid swarms. We speak of diverse populations arising from the meeting of two races which are interfertile, because otherwise they wouldn't mix. And we are more or less back to what we started with, depending on whatever was lost in the two populations. And again, this increase of diversity, the appearance of forms which are variable after the meeting of two races, is often presented as an example of evolution. That we can increase diversity by allowing populations to meet. Now, of course, we study nature, we try and repeat what happens in nature, and we try to use the natural processes for our own needs. Breeding. We breed plants, we breed animals for our own purposes. We select, but here the selection is quite often a positive selection. We don't select by eliminating what we don't want. We select for what we want. So, for example, if from this population we select for orange, this is what we'll get. We'll reject all the other individuals and we'll just keep the orange ones. And if from this population we select for blue, we'll get only the blue individuals. And of course, breeding works much faster than the natural processes, because we are eliminating many things simultaneously, just holding on to the individuals that are of interest for our breeding program. So we are getting races much quicker. And all these bred varieties are usually so depleted of genetic variation that they are helpless without us. We have to provide the environment for them. And the moment we stop providing the environment, this may be fertilizing, watering, giving a proper temperature, whatever. These are very delicate organisms. They are genetically weak. Most of the time, if we return them into nature, just leave them in nature, they will die. They will not survive because they are selected for the needs of man, selected for a specific type of environment. Of course, what we get will depend on what we're selecting for. And we know we have many races of dogs. And they look beautiful, and we speak of highly bred varieties or these races, and they often have a very high price and so on because of the specific characters that they have. But these are very weak organisms in terms of evolution. They are genetically impoverished, and they are very much dependent on humans to support them. Of course, if for some reason we forget about isolation, we'll get mongrels. We no longer have our beautiful races, we have a mongrel. This is because they will interbreed. I mean, there's no genetic barrier to interbreeding. The race exists as long as we maintain the isolation. And in nature, when there is a natural race, it will exist as a separate race as long as there is isolation. Now, conclusions. The loss of variation, and that also includes genetic variation. We speak of alleles being the genetic elements that get lost. This comes from, first of all, from isolation, because a fragment of a population does not have all its variability. This comes from inbreeding, because small populations inbreed, they homogenize, they accidentally lose genes. Then there is natural selection. Forms unsuited to a given environment do not survive. Thus, some of the genes get lost. When artificial selection comes in, when breeding, we specifically select for some types, enhancing the presence of some features and genes responsible for them, while eliminating others. Now, the main conclusion: loss of variation, loss of genes, does not give anything new. When isolation is discontinued, recombination of genes results. Some variants may be reconstituted, but nothing new results. And therefore, race formation is a process in the opposite direction to evolution, which requires new genes. So my first postulate is eliminate from all school textbooks the claim that microevolution is a small step in macroevolution, that formation of races is an example of evolutionary process. It is not. I've been hearing things like that the last couple of days. Examples are given of race formation, and this is advertised as evolution in action. Now, mutations. Let's go back to the original population. We know mutations exist. Many of them are neutral. When a mutation is neutral, it means that nothing has happened. Mutation took place, but it looks the same as it was before. So we don't see it, we don't notice it. It is a change that has had no effect. This usually amounts to the fact that the same protein is coded by a different triplet of nucleotides. That's for the biologists here who might know what I'm talking about. Anyway, in the genetic code, each protein has 3 nucleotides that identify it, but there are several triplets that identify the same protein. So if a change results that a different triplet identifies the same protein, we get the same proteins and nothing happens. We don't get any noticeable changes. When a mutation is injurious, this particular organism will disappear. We have negative mutations. We know they exist. And we know that nature will select against them because they're negative, because they are unable, the organism with such a mutation is unable to survive. So when we have such mutations that will eliminate something from the population, in fact, this doesn't affect the population as a whole. It can sustain losses like that. And most of the time, it doesn't have any influence on the population as such. Of course, it has an influence on the individual because the individual is injured and it may die or die sooner or whatever. Of course, we know that mutations are injurious. We fear mutations. We protect ourselves against mutations. We avoid X-rays, atomic experiments, asbestos, various mutagenic agents. We're not looking forward to mutations which will promote our evolution. We fear mutations because we know that they are injurious. They can hurt. So we do everything possible to eliminate mutations from our life. At one stage, there were big hopes that mutations could be used in breeding. Mutagenesis, this is called, the creation of mutants as a method in breeding. Some positive results were obtained, but positive in a sense of usefulness for us humans. For example, we got some dwarf plants, which could be used in rock gardens and so on, but in nature they will die. We have a variety of oranges that has no stones. The seeds are absent. But it's very nice to eat an orange and don't have to spit out the seeds. But nobody's going to convince me that this is a positive mutation from the point of view of the orange. It's not. It's a negative mutation. So there are a few results that have been useful or have been sort of justifying this type of research. But in spite of these so-called useful mutations, the mutagenic laboratories around the world are gradually closing, and I think most of those that I've ever visited are no longer functioning, because nothing really useful is coming out, and the cost does not justify the work involved. Mutagenesis as a method in breeding is no longer used, or it's only marginally continued here and there, but basically this is gone. In my field, in forestry research, I visited 3 laboratories that used cobalt bombs to create mutations in forest trees. All of these laboratories have closed this work. We haven't obtained any useful results for forestry. But I know in agriculture the same is true, that mutagenesis is gone. Now, what the text would say is that occasionally we have something new, something different. We have a positive mutation, something that would be selected for and not against. This is a postulate on which the whole of the modern theory of evolution is based, on the existence of positive mutations. Okay, if that is true, let's see some examples. And here we have difficulties because examples are absent. Now, a few explanations. What are mutations? They are accidental changes in the genes, in the DNA. They alter the existing order. Because nature is ordered. There's some logic in it. The mutation alters this order. They may have no effect, neutral. We know many examples. They may spoil the gene, cause it to malfunction or fail to function. We know many examples. We fear mutations. Now, improvements are postulated but not documented. And the only examples that are given are changes that bring resistance to man-made chemicals, antibiotics, pesticides, herbicides, and so on. Now, these changes that provide the resistance, they defend the existing order and functions. Most of such changes are achieved through recombination of existing genes. Recombination is the process in nature that allows to have variation. Recombination comes through sexual reproduction, and it comes through exchange of genes during meiosis, during reduction division. This is a natural process of mixing the genetic information that we have received from our parents, from the father and the mother, and each germ cell that is produced, whether it's an egg cell or a pollen grain or a sperm cell, whatever, it is a genetic identity of its own. It has some genetic information from the father, some information from the mother, and each is different from any other. Now, when these two germ cells meet and they form a zygote and a new organism, we have a new individual which is different from anything else. Unless there's cloning, division of the same organism, like in identical twins or in propagating by cutting a plant in two and getting two plants out of it. This of course, maintains the genetic identity. But basically, the whole biology is based on creation of diversity through sexual reproduction and through meiosis. So, this variation that is obtained through recombination may provide a variant which is resistant to one of these chemicals that have been introduced. Also, there is such a thing as immunological adaptation. This is actually a natural method, a natural defense method that organisms have to deal with alien bodies. Usually it's against bacteria or fungi or some parasite that attacks and is being dealt with by producing antibodies to deactivate it. So when we are given inoculations to defend us against smallpox or whatever, we are developing antibodies to fight possible invasion of some disease, some parasite. The immunological system is part of our biology. It is a defense method, defending the functionality of our organisms. And there are very few examples of resistance achieved by mutations. Now, one such example, which is advertised quite a bit, is this is a plant called Amaranthus hybridus. It is a weed. And a herbicide was developed called atrazine. The purpose of this herbicide is to kill Amaranthus. It is a specific herbicide against a single weed. Now, the atrazine was developed to attach itself to one of the proteins. The protein is called QB, and it is coded by gene PSPA. Now, the atrazine QB complex, when it forms in the plant, prevents photosynthesis, so the plant dies. So atrazine is a herbicide that kills only this species, or related species, but anyway, it is a weed-specific herbicide. There is a mutation that changes a sector of the DNA which defines a segment of the protein QB, by changing one amino acid, serine. Serine is coded by the triplet AGT, adenosine, guanine, tyrosine. And when this triplet mutated, with the A mutating to G, we get a GGT triplet, which codes glycine. And in the protein in question, instead of serine, we have glycine, a different amino acid. The protein is still functional, but it has lost its attachability to atrazine. So the protein functions, and the plant survives in an environment where atrazine is used. So we have a mutation here, this mutation, A to G. This is in a specific position, in a specific gene. We know all the details. And we know that this provides resistance. And this is proclaimed as an example of a positive mutation of evolution in action. There is a need of a change in a specific environment. This change was produced by mutations, and the plant survives. Now, the evolutionists conclude mutation of one nucleotide from A to G gave resistance to atrazine. This resistance is considered a new function. This example advertised as evolution in action. Now my conclusion, critical conclusion. The change defends the functionality of protein QB. It defends its role in photosynthesis. No new function produced. This is a defense reaction. The change is at best a neutral mutation. It maintains the function. The protein with the glycine instead of serine in one location is still functional. Actually, in atrazine-free environment, there is selection against this variant. So for some reasons, the original one is better and nature prefers it. But as long as atrazine is there, this variant dominates and it defends the wheat. The protein variant with glycine, that is in the DNA with the GGT triplet, is less adapted because it is selected against in a natural atrazine-free environment. So the mutation is slightly negative, or we can call it neutral. Now, as I see it, evolution requires introduction of new functions and organs. Neither race formation nor mutations produce new functions or organs. And textbooks that speak of race formation, or microevolution it is called, that speak of adaptation to antibiotics or herbicides, that speak of positive mutations, giving resistance, as examples of small steps in evolution, are misinforming. Thank you.
My only doubt is, you said at the end, final conclusion, evolution means developing of new organs. Because that, I'm a bit puzzled by that, because as you also gave an example of a hybrid, what about the hybrids in the case of plants and crossbreed in the case of animals, for example dogs, artificially done? Won't you consider that as a step forward, as some sort of a positive mutation? No?
Well, first of all, it's not a mutation in the sense that, the word is used nowadays. It is a change, if you like. But the point is this, that if you have something new produced from crossing two types, the variation, the genetic information already exists. You are just producing a new combination of that genetic information. I might add another thing. This is something that is very much being studied right now. This is what is known as horizontal transfer of genes. We know that they can be transferred to sexual contact, but for that they must be interfertile. But in nature, genes are also transferred from one organism to another by other methods than sexual. Now, we have learned to do it by studying nature, and we are doing this, and what we get are what are known as the GMOs, the genetically modified organisms. Because we will transfer a gene, say, from a fish to a tomato, and this tomato will have some property, for example, of resistance to freezing during transport, because the gene from the fish is resistant to low temperatures. And so the tomato is on the market, and it has been long time in transport in the freezer, and it doesn't wilt or whatever that would normally do if it were held in a cooler without that particular gene. So we transfer genes horizontally between organisms that are completely unrelated. You can't cross a tomato with a fish, but you can transfer the genes. You are producing new things, but these new things are based on information that already exists in nature. We are just using what already exists. And such horizontal transfers occur in nature naturally. We are just copying what nature does. Now, you are speaking about extinction of various organisms. Yes, indeed, we observe extinctions. we are worried that the genetic diversity of the world is declining. We try to protect genetic diversity. We try to protect animals that are close to extinction. And of course, there were extinctions in the past. We see it in the paleontological record. There were organisms which lived on this earth, but live no more. Some we remember because people have seen them, like the dodo. There are some animals or plants or birds that have existed, but no longer exist because for some reason they have disappeared. Now, what has changed is the proportion of animals of different types. We know that in the past there may have been a different proportion. There may have been some animals which no longer exist that were quite important, that they played a significant role, and now they don't exist. So in their place, other plants or animals have filled in the niche, the ecological niche that was occupied by those forms which are already extinct. But that is no argument for evolution, because we are not getting anything new.
Thank you for your presentation. You spoke about the effects of isolation and genetic drift on populations. Have we witnessed situations in which two isolated populations, when brought back together, were not able to interbreed because of the level of distinction that had developed over time, so that they were no longer able to interbreed when reunited?
Quite often it happens that we assign species rank to populations which we consider as not interfertile. It is possible that they have originated from the same population originally. In my field, forestry, there is such a thing as European larch and Japanese larch, and they are considered as two different species. Now, Japanese larch was introduced into Europe, and it is interfertile with the European larch, and we get hybrids. And in fact, quite often people are worried, well, this is affecting the cleanliness of our nature and so on. We should prevent this foreign species from changing our... Quite likely, the larches have separated at some glacial period before and so on, and they have moved so far apart that we distinguish them from descriptive methodology in systematic. We have described them as separate species, but perhaps this is not justified. Perhaps this is the same species. Now, to answer your question more specifically, this is known in breeding of animals. Now, you can imagine that there are races of dogs which are so different in size that they would not interbreed. But if you do in vitro fertilization, you'll find that they are interfertile. But in vitro is not happening in nature. You know, this is. So through breeding, possibly through natural divergence of populations, perhaps such differences could develop that would make compatibility impossible. And this, it is what often happens is that we have in these races that have separated, that they are adapted to different climatic conditions, and as a result have different timing of reproduction, time when pollen flows or the flowers are receptive and so on. So we put them together and they don't interbreed because they flower at a different time. But if we humans keep the pollen and then use it to pollinate and so on in an artificial manner, then we're getting hybrids. So the definition of species is man-made, and we may have to alter it as science progresses for specific organisms.
I was very happy to hear from you that there is so much misinformation in the school curriculum to the students, a false picture with regard to evolution and mutations. Now, I'm asking whether is this body or you really demanding the revision of this kind of teaching from the school, I think.
Well, this is what I'm trying to do. This booklet that is being distributed right now, I have produced it in the European Parliament, where I am a member, and I tried to get some reaction. Of course, the reaction was negative, and I'm very severely criticized for being so unacquainted with the modern science that I'm presenting things like that. But the interesting thing is that nobody is picking up a polemic with me. I'm being attacked for being a religious fundamentalist, which I'm not, because there was a time when I accepted evolution, being a Catholic all the time. I mean, I can quite accept the suggestion that God was creating through evolution. But as a scientist, I see that evolution has no scientific basis. So it is not my faith that is the problem, it's the faith of the evolutionists, who are insisting on evolution in spite of the fact that they don't have any evidence for it.
To close the discussion, could I ask you to give us your opinion about gene therapy that you are using in medicine at the moment? What is the limitation of the process in your mind?
Well, of course, I'm not a medical man. I know that gene transfers are being used quite a lot in the production of pharmaceuticals. So, for example, insulin today is produced by some, I don't know, bacteria or fungus or something. This is a trade secret. It is a patented thing. But this organism, this microorganism, has been enriched by a human gene for the production of insulin. A good human gene, not devoid of any mutation, spoiled by any mutation, but a functional gene. And this stupid bacteria of fungus is producing insulin that it doesn't want. And we just collect it and use it for the injections that doctors prescribe. Now, currently, there are attempts at planting a gene with the correct genetic information to a patient who has a defective genetic information, and this patient with the gene from another person, but without the defect, sitting on a virus that is neutral but will multiply in the organism and will provide the possibility of producing whatever the patient was unable to produce himself. That's gene therapy. Now, as any new technique in medicine, this has to be tested where there are no side effects, some negative, some problems with which might be collateral to the actual intent. So these are new things, and I don't know whether they have reached any stage of beyond clinical tests. I'm not a medical man, I don't follow this sort of literature, but there are hopes associated with this type of research.
It's true that it's a hope, but it's some limitation you could observe. We observe at the moment some malformation or defect after gene therapy. That means it's probably a difficult joke.
Well, before it can be used on a mass scale, it has to be tested that it has no negative effects.
Okay, thank you.